presentation will be recorded. We'd like to start with recording now, so it will be available. Presentation's over. I'm joined on the line today by Richard von Furstenberg from the University of North Carolina, Chapel Hill. We'll be discussing about side population analysis and CD24 analysis in intestinal stem cells. Before we start, I'm going to be focusing on stem cell enumeration specifically. So today's presentation is going to focus on two different applications that we can use in stem cell type assays. We're going to be looking at a clinical assay, stem cell enumeration, and then we'll be talking about research applications for stem cell assays, and that's what Rich will be focusing on. So we'll start talking about stem cell enumeration, and just as an introduction, stem cell enumeration is a clinical application where it's an assay that's used to help detect and quantify CD34 hematopoietic progenitor cells. The idea being that the enumeration of these viable CD34 positive cells per kilogram of recipient weight has been shown to be a very useful indicator of whether or not a graft success has been achieved within a patient after myeloablative therapy. Specifically, these patients are going to be apheresed, and when they're infused, they typically like a concentration of about 2.5 to 5 times 10 to the 6 cells of viable CD34 positive hematopoietic progenitor cells to reliably predict the successful engraftment within the system by day 12, day 14 post-infusion. And there have been studies that have shown that a higher concentration of infusion can yield some quicker results in as little as 8 to 10 days. Prior to flow cytometric assays, CD34 was previously counted through colony-forming unit assays. The problem with these assays were that by the time you can count these colonies were already lineage committed, whether it was to granulocytes, macrophages, whatever. And in the early 90s, the first flow cytometric iteration of a stem cell enumeration assay was developed in Italy by what is now known as the Milan Protocol by Salvatore Siena. And it was essentially a 3-parameter assay where you're looking at forward scatter, side scatter, looking at all your events above debris, and then taking those events and looking to see what was CD34 positive or so. Now, the issue with that was that it wasn't overly specific because there were some non-specific events in there. And after the introduction of the Milan assay, there were other variations of the assay that came along, specifically assays like the Sihon method developed in Europe, the Bender assay, which eventually introduced the idea of having CD45 in there to further specify these cells. But in 1995, in late 95, there was a group called IceAge that introduced a stem cell enumeration committee, where the idea was that they'd like to provide a simple, rapid, and sensitive flow cytometric assay to quantitate these CD34 positive cells in peripheral blood and apheresis products. The method was introduced in '96 in a paper in the Journal of Hematotherapy and Stem Cell Research by Sutherland Keeney et al. And the method was that it was an idea where you can look at cells and further classify them according to fluorescence specificity as well as scatter specificity, whether it's forward scatter relative to size or side scatter relative to nuclear complexity. The introduction of the IceAge method has been proven to be a reliable source to help standardize assays amongst results. Prior to the IceAge method, there were different iterations of the assay where performing the same assay at various sites kind of yielded different results. So the IceAge method provided a good way to help standardize this assay among different sites. And the whole idea behind the IceAge method was that it was a hierarchical system where you could focus on your cells of interest while limiting the impact on the counts from the non-specific events. So you're basically looking at cells, you're taking these cells and then you're breaking it down according to fluorescent sensitivity and specificity, looking at CD45, CD34, and then some scatter properties. Now, the method has evolved over time. Because the method was developed on a sequential Boolean gating method, it really lacked the need of an isotypic control, as they found out that by further focusing on these drilled down populations of CD45 cells, CD34 cells, DIM45, and focusing on scatter, it was really easy to pick out these populations, these clusters that were available within these samples. It also gave you the capability to quantitate these cells either through a dual platform method or ideally a single platform method where within the sample there would be a counting bead that was available where if you count these certain beads then you can use an algorithm to help decide what was the overall concentration of these cells per microliter. And then also it gave you the ability to use a viability dye such as the DNA stain 7AD. 7AD seems to be the most popular, though I have seen labs that have used something as something like Topro, where they were able to set it off the red light. And down below is a graphic showing the Sutherland McKinney article. And also just throwing out there, 100 bonus points to the first person who can enter into the questions section what ICH stands for. Honor method, no one Googles it. 100 bonus points. Okay. So from here on in, what we're going to do is we're going to focus on the analysis specifically. So here we have this template. There's a lot going on in this template, so what we're going to do is we're going to first look at some of the features within FCS Express that would help us analyze this data reproducibly and easily. So today's webinar is not really going to go through the basics of how to create these things or how to insert some of the basic objects. But what we're going to do is we're going to show you some of the highlighted specific features that can help make an analysis like this for rare events, because that's what CD34 is. It is a rare event. How we can build some of these, what we're going to call tokens, within this assay to give us numbers that are going to be specific for our experiment. So the first thing I'm going to do is I'm going to highlight a feature within FCS Express called panels, where we can make these analyses, these layouts, panel specific, based off of a set number of tubes. All we need to do is load our tubes into something called the data list. And because we have this as a panel-specific assay, specifically for four tubes, once I select my first tube of my 4-tube panel, FCS Express can automatically import the next 3 to complete the 4-tube assay. Right now you'll see that this is a 6-page layout that we have here with various elements, analysis elements throughout the 6 pages. And if I click yes here, the tubes that are in the folder will automatically load into my layout, into my data list, and then you'll start seeing the data and the numbers automatically appear on the various pages. Okay? So within the data list, you can see here that using a panel, what we can do is we can give a descriptor as to what constitutes each tube within our panel. So here my panel is made up of two levels of control, a low control and a high control, and then we're basically taking a patient that we run duplicate. And what we're going to do is
welcome to GoToWebinar, Web Events Made Easy.
Of the type of analyses and samples that we're looking at. And we can easily add keywords to this list just by clicking on the Format button here in our data list and just adding keywords. So if you needed something to give you an indicator of the type of sample that we're using, or maybe you just want to see what the patient name was, or whatever kind of keyword to save with the data file, you can actually add it here in the data list so you can see it automatically. But let's focus on the layout specifically now. So the layout now, as you saw before, was 6 pages, but now as we loaded data, it's now trimmed down to 4 pages. So I'm actually using FCS Express 4 Clinical Edition. One of the features in FCS Express 4 Clinical Edition is that we have the opportunity to hide pages that we don't really need for analysis. In this case, because the first 2 pages were just text, it was just basically part of my presentation. Once I loaded the analysis in, I actually told FCS Express to hide those once I loaded my data into my plots. So now I can actually focus on the analysis, but I still have those. I had those slides for the presentation there prior to presenting the data. Now, if I click on my controls page and my raw data page, we can actually focus on the raw data, and we can see what's going on here specifically. So if I just look at my raw patient data page, here we could just talk about the gating strategy that's involved here in an ICH method. So up here, what we're doing is we're trying to focus on our target population. And since our target population are these CD34 positive hematopoietic progenitor cells, we know that these cells are going to be moderate to bright intensity of CD34 expression. They're going to express a dim DIM45 expression. They're going to be fairly low in side scatter, and they're going to be moderate relative size, roughly the same size, relative size as lymphs, or maybe just a little bit bigger. Okay, and that's what this gaining strategy is actually showing here. So first of all, we look at this plot. What we're doing is we're gaining our beads, and we're taking those beads and we're actually removing them. We're counting them, but the rest of our analysis is going to focus on everything but the beads. All right. So if we look here, we have something called our gate view. And our gate view is going to show our relationship of all our gates and all our populations to one another. So here we have our beads. Here we have our whole cells. So what I did was I created a Boolean gate or a combination gate within this gate view just by right-clicking and adding a combination gate. And if I double-click here in this population in my gate view, it's going to show me how I created it. So basically I just told the software that I wanted to define this whole cells gate as everything that's not a bead. And then I added a color to that. And then I'm looking at these whole cells within the CD45 gate, and I'm getting these CD45 positive cells while also trying to remove all this debris down here. So it's important to remember that with an assay like this, it is considered a light snow wash assay. So whatever's in there is in there. We don't really wash it out. We don't do anything with it. So assays like this, it's very typical that you're going to have a threshold on something like CD45 to remove as much debris as possible. So because of that, we've removed a lot of our debris, but we're also going to remove this debris that's here and just gate around it. Because if we increase the threshold anymore, chances are we're probably going to remove some of our cells from our analysis, and that's not something that we would really want to do. So we're going to look at these CD45 positive events within this gate and look at CD34 positive expression based off of these CD45 positive cells. So you can see here in the title of these plots, it's looking at specific populations. Okay? So now I'm taking these CD34 positive events and then looking to see which of these cells are actually a DIM45, just like we are defining these cells over here. Once we take these cells, then we're going to look at scatter. Now we would expect these cells to be in a nice tight cluster on forward side scatter. Anything that's lower than that is probably debris or maybe just some type of fragment of a stem cell. We don't really want to care about that. We don't really want to count those, so we just remove that from the analysis just by gating on that nice tight little cluster. If I go back to my hierarchy here, you'll see here that the hierarchy reflects that my stem cells are actually going to be a subpopulation of my overall whole cells. But what I also did was I have a viable gate here that's based off of my whole cells where I'm looking at 7AD. So anything that's 7AD negative, meaning that the DNA dye 7AD hasn't had a chance to penetrate the cell because the cell's not permeated, it hasn't entered apoptosis, or it's not necrotic. The dye has not been able to penetrate the cell and attach itself to the DNA. You might get some expression, but overall you can see that most of these cells are viable. In FCS Express, what we can do is we can just create a little statistical token where we get a quick idea of what the overall viability is. But what I can also do is I can combine these two gates where I say that I want to look at the cells that are both in my viable gate and in my stem cell gate and create a new gate off of that I'm considering my viable stem cell gate. If I double-click on that, then you'll actually see here, just like we did before with the whole cell population, we can see the formula that I used to create that gate. And I also put in a comment here saying that the cells of this gate are the cells that are in both my side scatter, my stem cell, and my viable gate. Right? So once we tabulate all of these stats, what we can do is we can now calculate an absolute count. So down below here in a text box, I just put in some text showing you what the overall formula that's involved to calculate these events. And with this particular product of counting beads, we know that the formula is going to be the number of events counted versus the amount of the bead count that's in that tube. With this particular manufacturer that provides these beads are a lyophilized bead product that's in a tube that we already know what the overall count is within that bead product. So we can take that number, because it's actually listed on the pouch that these tubes come in, and then multiply it by any sample dilution factor that was established for the sample. Now, typically for these kind of assays, you're going to have a linear range for concentration of your samples. For this particular assay, your cellular count needs to be about 40,000 cells per microliter or less. If not, it's preferable that you make a dilution factor out of that and then enter the dilution factor into the calculation here. And then we're going to take this quantity and divide it by the number of beads that were counted overall. So for example, the 7,003 beads that were in the bead gate multiplied by the sample volume. The sample volume in this case is going to be a constant. It's 100 microliters. Okay? And then you'll see here that we have a text box where we entered what we call tokens. And what tokens are, they're dynamic text that are going to update according to the data that's in your analysis. And it could be as simple as something like the amount of bead events or my percentage of CD45 cells. Or what we can do is we can actually use tokens in the creation of what we call custom tokens. So if we wanted to have a mathematical formula represented in our analysis, we can make a custom token where we enter all of these individual statistical tokens to give us an overall product. And that's what this number up here is, the 30.52 cells per microliter and the 20.97 cells per microliter, is using each of these samples using this formula down here. If I double click on one of these, it'll show me what the overall formula is. So for example, if I look at the value, I'm looking at the number of events times the bead count divided by the number of beads counted multiplied by the sample volume. Okay? So this is essentially the same sample that we ran twice. So chances are we just probably want to make sure that the number of stem cells that we count here correlate with one another. So down below here you'll see I have a little text box where it says viable CD34 cross-checked. And basically what I want to do is I want to perform some type of statistical analysis to see whether or not these stem cells correlate with them in each other, specifically a 10% correlation. So when we create custom tokens, what we can do is we can create something, a custom token using something called a function. And this specific function that we're using is a function called R values in range, where we put in some numerical data, whether we type it in or we actually refer to other tokens. We put in the tolerance, meaning how much of a percentage do these numbers need to check. And what the textual output is, what the textual output is going to be if it turns out that these are not within 10% of one another. When we create this type of token, what we can do is we can further use it in an if-then-else statement where if it turns out that if this is true, then we're going to write the word true, or we're going to write the word OK, or we can actually have it reflect back to another token. So for this particular token, what I'm saying is that if it turns out that the CD34 from this tube is within 10% of the CD34 of this tube, it's going to give me the word OK. Otherwise, it'll tell me which one's out. Okay. Also, I want to show you the capability in FCS Express to create a bar chart. So a bar chart is a really good way to show you a quick snapshot of what's going on with your overall sample, where we can create a bar chart just like you would create a bar chart in Excel. And I'm not really going to go into this into too much depth. Rich's presentations will show you bar charts that he's created for his experiment, and he'll be able to talk about that a little bit more and show you specifically what he's looking for in his experiment that he can get across with the bar chart. But what I'd like to do is I'd like to focus a little bit on our QC and patient summary page here that we're using as a report. So on this page, there's a lot going on. I just want to highlight. So here on this page, we have a number of different tables, and each one of these tables has a purpose that I've incorporated into the analysis. And all these tables is I've inserted a text box. Within this text box, I've inserted a table. And that table can be as big as you want it to be. You just have to designate how many rows and how many columns. And then within each of these fields, we can put in information. We can also edit these text boxes to merge cells, we can split cells, we can add shading, just like you see here. So each one of these tables is going to be specific information that we're providing about the samples that we're analyzing. So what I did was I'm using various types of tokens that we can extract from our information and provide it within these tables as a way to summarize what's going on in our analysis. So if I look here in my sample information, it's basically a table that's summarizing demographics. So what I'd like to know is my patient name. I want to be able to summarize what the sample ID, if it had the sample ID to it, when this particular sample was acquired, and who did the analysis. If I click within this table, you'll see that some of this is just regular text that I typed in, and I can bold this and edit this through our text editor. And then some of this is actually tokens that we're extracting from various data. In particular, some of these are keywords, where we're able to add a keyword token and then look in the FCS file header to extract the value of a particular keyword that's being used in the analysis of that data file. All right. So just by right-clicking one of these plots, we can add a token, choose a specific type of token that we're using here, and then we can choose the type of keyword that we want to enter. If you're not familiar with keywords, you'd be surprised the amount of keywords that are available with you within your data in the FCS file header. And a lot of these keywords are going to be specific to the type of software and instrumentation that you're using. If you need help with keywords, just let us know. We'd be happy to help you with that. Also here I have some tables looking at QC and also a summary of our data. Specifically, I want to look at the QC here specifically because the QC is actually pretty interesting, because what we have here is we have tokens that are giving us a range, and it's telling us whether our QC value is in range or out of range. So if we look at the range here, what we did was we created custom tokens where we're looking at what the overall mean is, and then the mean for our QC values are provided to us by the manufacturer. And so what we're doing is we're taking that mean and then we're just developing a range that's 20% below and 20% above whatever that mean is. We're using that range in the evaluation of this particular value, and this value is just the overall stem cell count for our controls. And you'll see here that there's a token here that's called in range. And what this is a token that's specific to our clinical edition called the statistic classifier. If I double click on this, it'll show me the classification that I'm using. So specifically what I'm doing is I am classifying a particular token, and what I'm doing is I'm looking at the value of that token and determining whether or not it's in range, or out of range. And I'm using the values that are determined by this 20% below and 20% above cutoffs that I've created as a way to evaluate whether or not those values are in range. And if it turns out that this number is low or high, then you'll see that the status will change to out of range. And what I'm going to do is I'm going to see if I can trip that. So one of the things we can do is we can look at multiple pages at once. I'm going to go to my control page on one page, scroll this over here. And now that's really low, so you can see here that it gives me that output that it's out of range. So that's a very useful tool that you can use within your analysis. Overall, we have an overall summary chart looking at the various numbers that are being produced here, where we're taking, again, that custom token that we use to calculate our absolute counts, and we're doing based off a total and also viable. And then what we're doing is we're using another function where we're looking at these two values and establishing a mean. Okay, so we're doing that right across the board here for each of these particular values for the number of beads, the total, the viable, and we also have a token here for the percent of viable cells per CD45 events. And then we're also doing a QA check to determine whether or not those are within 10% of one another, just like we did earlier with the R values in range custom token that you saw me talk about earlier. Also here, what we can do is, one of the features of the clinical edition is we can insert something called canned comments. And what canned comments are, they're predefined comments that we can create within FCS Express, and we can create this library where we can have FCS Express extract these comments and just put them into a text box just like we see here. So from the view menu, you'll see that there's a tool for canned comments. I can just click and drag this in here. Not sure what happened there. But what we can do is the canned comments, we can click and drag into our layout. And a canned comment could be anything. It could be regular text. It could be tokens. We can insert tables. We can also insert canned comments in here just by right-clicking and just adding them through the drop-down menu. So these can be as simple or as complex as you need. But specifically on this page, what I'd like to do is I'd like to focus on the fact that CD34 overall is a rare event. Because it's a rare event, it's important that we apply Poisson stats to this type of analysis. So in other words, the standard deviation is the square root of the number of events that are counted. And because the consensus range of CD34 positive cells in peripheral circulation is about 0.01% to 0.1%, this classifies itself as a rare event. So following that idea, for a CV of 10% or less, we're going to need at least 100 viable CD34 events being collected, as well as 75,000 CD45 events. And I've also thrown in a criteria here just to make sure that 1,000 beat events are collected here. So what we've done is we've created tokens where we're evaluating whether a particular condition is true. So I've created a custom token just to look at my CD34 events and just to make sure that it's greater than 100, just by looking to see whether or not the number of events within a gate is greater than 100. If it turns out it's true, then the custom token that I made here is going to give me the output yes. If not, no. And I've done that here as a safeguard to make sure that my assay, my overall assay, is as precise as possible. Okay? So these types of analyses that you can perform within your assay are very critical in this type of rare event analysis. So the ability to do this, there's a lot of value in that. And the idea that this can be reproducible should underlie that concept. Okay? So I know that might be a lot of information up front, but the idea is that we'd be happy to help you do this if you need the help. You can always contact us at support@denovosoftware.com. I'd be happy to help you. At this point, I'd like to pass the mic to Richard von Furstenberg, who's going to talk about side population analysis and CD24 expression in intestinal stem cells, and also as a brief bio. Richard is a research specialist at the University of North Carolina at Chapel Hill. He received his BS in marine biology from Texas A&M and obtained his master's in cell and molecular physiology from University of North Carolina while working in the lab of Susan Henning. The research performed by the Henning lab is focused on isolating and characterizing adult intestinal epithelial stem cells so they can be used in regenerative medicine applications. Richard's projects in particular involve side population sorting, as well as surface marker approaches to isolating intestinal stem cells by flow cytometry. Okay? So with that.
Hi, yeah. Thanks for the kind introduction, Juan.
Sure, no problem.
Somebody can message me just to confirm that they can hear me okay. That would be great. And I'll start.
So I'm just going to put on our side population template. And it seems like we do have a question.
Great.
Yep, it seems like everyone can hear you.
Great. So first I want to kind of give you guys an overview of the intestine for those of you that aren't familiar. And our, like Juan said, our interest is in stem cells, and it's not only in isolating them, but if you come full circle, it's how we can manipulate them in vivo and possibly transplant them as an end goal. But that's kind of working backwards. In order to understand how they work and to transplant them, we have to isolate them to understand how they function. And so taking a step back, looking at the intestine, it's essentially a tube that serves to absorb nutrients and protect us as we invite the outside world into our body. And so it does this very capably, and it uniquely has a very active stem cell population. So part of this protection is to have a very rapid turnover of the epithelium inside of the intestine. And here you can see on the left this kind of description of the tube, and you can see a piece splayed out. And then in this inset, you can see these finger-like villi that aided absorption by increasing surface area. And at the base between these villi is an intestinal crypt. And what makes this tissue difficult to work with are these tight junctions that help protect. So with bone marrow isolations, you typically break the femur if you're using a mouse, not a human, and squirt the cells out and collect them, and you kind of have them in suspension. So the intestine, one of the benefits is that we do have this rapidly cycling stem cell population that allows our epithelium to kind of turn over and dump out in feces every 3 to 5 days. So if a cell becomes corrupted, it's just tossed out. And it's a wonderful evolutionary solution to invaders. But yeah, we also have these tight junctions that keep bugs from crossing the intestine-endurant body, and these tight junctions make it difficult to isolate. So if we can go to the next slide here. What we're doing is we're zooming into, in this inset, a Crypt Abase that I described earlier, and this is where the progenitor cells rise from. You can see the stem cells shown in orange down at the base of the crypt, and they're intercalated amongst Paneth cells shown in green. And Paneth cells are just one of the lineages, the 4 lineages that arise from these stem cells. These Paneth cells serve to protect and also as a nurse cell for the accompanying stem cell. These progenitors, shown in yellow, migrate upwards and take on one of the 3 villus lineage fates: enterocyte, which absorbs water and nutrients, a goblet cell, and they secrete mucus, which protects, and enteroendocrine cells, which communicate to your brain what is going on inside of your gut. So your brain is kind of constantly in touch with what's passing through you and can respond appropriately. So it's really an amazing tissue. And it's been known for more than 30 years now that the intestinal stem cells reside in the Crypt Abase, and this was early work using tritiated thymidine in the 70s. But we're way, way, way behind bone marrow. So it was only in 1996 that there was kind of some headway in the isolation of intestinal stem cells, and it's progressed somewhat slowly from there, although it's recently picked up because of the use of transgenic mice in the intestinal stem cell field. So at the time that the first headway was made in 1996, there was really no membrane marker approach that was viable. And Peggy Goodell, who was doing her postdoc at MIT, along with her colleagues, developed this method using Herx 33342, which is a DNA binding dye. And it was a serendipitous discovery. She was staining bone marrow cells for DNA content and noticed that there was a small fraction of these cells that wouldn't absorb the dye. And this percentage happened to match up with what they expected for the percentage of bone marrow stem cells. And so this became kind of an alternative to a surface marker approach. So as I mentioned, this was a very fortunate discovery, and it relies on the efflux of the vital dye by membrane proteins unique to stem cells. And so this seems to be a conserved mechanism that these stem cells express these effluxing proteins on their membrane, and these kick out not only dye, but also any other kind of xenobiotic invading molecules. And here on the left is a plot of what a side population looks like, with Herx blue on the y-axis and Herx red on the x-axis. And then the SP is gated here with the blue gate. It kind of looks like an apostrophe tail kind of coming off of the population. And so the Herx 3342 is a DNA binding dye. It binds to the narrow groove of the DNA. And at low concentrations, it emits pretty much only in the blue channels. And with enough of the dye, as that peak rises up, the nonspecific emission of a red wavelength light also occurs. And so if a cell is saturated because it cannot efflux this dye and doesn't have these efflux transporters unique to stem cells, it takes on a red shift, and that's this kind of blob in the dead center of the plot. And these are the non-SP cells. And what we're looking at here is an overlay of a side population in red. This is with Herx dye, and a verapamil plot in black behind it. And so verapamil is a calcium channel blocker that we use to stop the efflux being carried out by these efflux proteins. So we're basically forcing the SP to become non-SP as a validation that it is what it is. And that way we're not just gating this apostrophe-looking population and hoping these are stem cells. And this overlay is a very nice feature of the FCS Express 4. Traditionally, what I've had to do is take these two plots into Photoshop and create layers and make one transparent and put it over the other, and it's a real pain. So I'm really happy that this function is here. because sometimes you want to track multiple plots and backgate them to one source plot and overlay all of those. And so this will be very useful. Subsequently, it was found that this SP sorting worked in many tissues, and in 2005, a postdoc in our lab used it on intestine. And if we can go to the next slide here. Interestingly, we did find a side population in intestine. We were really excited because this was the first method to isolate intestinal stem cells from wild-type mice. And what that allows us to do is also have a technique that could carry over to human in the future. And so what we're looking at on the very left-hand side is that first side population plot overlaid with a verapamil. And now what we'll do is kind of walk through the gate logic. So we start out with this prep that's very heterogeneous. And I had mentioned there's 4 lineages in the intestine, and each of them are very different in their characteristics. Some are, big and fat and round. Some have granules. Some are small and agranular. So you get a very heterogeneous mix on that forward scatter, side scatter plot. You might be used to bone marrow where you see kind of very defined clusters, but that's not the case for us. So with side population sorting, we don't want to be too discriminating there, and we kind of let everything through. And then we say, well, what we want first are singlets. And so we select based on pulse width, and we take those singlets and carry them forward to a CD45 gate. And here what we're doing is removing any contaminating bone marrow, cells of bone marrow origin, rather, because what we don't want are bone marrow stem cells getting into our prep and growing and giving us a false impression that we have intestinal stem cells, generating some tissue. The next thing we do is we take those CD45 negative cells and carry them over to a PI plot. So we're using propitium iodide to select for live cells. And the cells that take this dye up that are permeable shift to the right, and we discard them. And so I'm sure this looks pretty striking to a lot of you that only 20% of our cells are alive, but rest assured this is normal with SP sorting. Depending on the concentration of HERCs, you get roughly 20 to 40% of the cells that live. And that's because of 2 things. One, the dye is somewhat cytotoxic. And 2 is that if the incubation time is an hour and a half at 37 degrees, and so these stem cells seem to survive the anoxia, while a lot of the other lineages don't. And so the process itself is somewhat of an inherent selection for stem cells. We then take these live cells to the next gate, which is the actual HERCs plot. So here we're looking again at the HERCs blue by HERCs red, and the SP is shown here, gated in green and highlighted by yellow. And so other vital dyes can be used. Some papers have used rhodamine 123. We ourselves have also used dicycle violet on some of the cyans. You can use the 405 and move the filters around and generate a side population, but still nothing quite gives us the resolution that this UV and HERCs combination does. So all the stats from these gates you can see are down here in the panel below. And so the calculations for the CD45 negative, live cells, percentage of side population cells, and all that data is tabulated for the HERCs sample, a verapamil sample, and a post-sort sample, and we'll walk through those. So the nice thing is that as these gates move, the values change. And here we're looking at the verapamil sample. So these are the same cells, a homogeneous prep of intestinal epithelium, but this time they're pre-incubated with a verapamil to shut down the efflux of the stem cell. So now they're forced to take up the dye, and you can see that they've all cleared out of the SP gate that was pre-established, and they've shifted into this non-SP bunch kind of in the center of the plot, validating that what we have is in fact side population. And in the bottom right, you can see the gate logic lined up. And Juan showed that earlier with the human cells. This is a post-sort of side population cells. So this is extremely important when we're doing downstream assays. So when we collect these cells to do qRT-PCR or RNA deep sequencing, we have to be super sure that what we're collecting is what we think it is. And the way we do that is by taking some of what we've collected and running it back through the machine to make sure it falls in the source gate. So here you can see that 97% of these cells track back into the SP gate, and they are singlet, and they are CD45 negative and alive. And so this is an extremely important validation whenever you're doing any kind of downstream analysis, just to double check that what you think you have is what you really have. To make this data interpretable to people that aren't familiar with side population sorting, or if you don't have a lot of time to convey the ideas, putting in the data into this bar chart is really handy. Here we can see the three samples, the Herx, the verapamil, and the post-sort, and we can see the values for live, CD45 negative, and the percentage of SP cells in each one. So I find this very beneficial for getting this data into a format that's quickly understandable, and it happens seamlessly. As you're moving your gates, these values change automatically. So there's no need to take your data, move it to Excel, and then make these plots. Juan has showed me that we can move these, we can actually export these into an email or into a PowerPoint. And so that makes communicating these data very straightforward. So one great product of our subpopulation sorting was the fact that it gave us candidates for a surface marker approach, because obviously a surface marker approach would be more gentle than this harsh dye incubation with the cytotoxic dye, the Herx 3342. So what we did is we took side population cells, which we knew by this time were enriched for stem cell transcripts, and crossed them with a laser capture microdissection of the Crypt Abase, where the stem cells reside. And this capture from the Crypt Abase, where the stem cells reside, as I described, it has panel cells down there in the Crypt Abase, and we don't want those. So we took this laser capture from a mouse that didn't have panel cells, and we crossed it with a side population. So although they both have contaminants, they don't have any contaminants in common, or that was our thinking. So we did the crossover of these transcripts and found that we had 358 potential ISC transcripts, or intestinal stem cell transcripts. We then performed gene ontology analysis on these transcripts and found that we had 14 that had a presence in the plasma membrane of the cell. And 6 of those have nectodomains. And the one that was most promising was CD24. And this was published, this was a paper published by our lab in 2011. The citation is there in the bottom right if you want to look it up. What we found was that indeed CD24 is a marker in intestine for stem cells, as it is in other tissues, in memory tissue, in brain, and others. And if you're looking here at this top left panel, this plot A is a low power shot of, this is a cross section of intestine. You can see some of these long villi stretching up. They're a bit irregular. But what's pretty uniform are the crypts down at the base. And you can see this kind of concentrated brown staining. Yeah, panel B definitely shows it more clearly. You can see a crypt here, and you can see the dense CD24 staining at the very base in the stem cell zone. And so what you'll also see are some kind of brown hot spots in the endothelium, which are these, it's essentially the villus core. And we're not concerned with that because we make an epithelium preparation. So we're only taking the cells from this crypt base that have CD24. What we found in the bottom left panel here is that we have the published marker LGR5. So Hans Clevers has a lab in the Netherlands, and they came up with this great transgenic mouse, the LGR5 EGFP high mouse, or EGFP mouse rather. The cells that express EGFP high at high levels are the intestinal stem cells. And this is great for a tool for investigation, but as far as having a marker that's relevant to wild-type mice or human, you need it to be universal. And so that's why we're pursuing CD24. But LGR5 in this reporter mouse construct gives us a great tool for validation. And what we found was that 95% of these LGR5 EGFP high cells fell in our CD24 low. So this gave us a great validation tool. However, the mouse is mosaic, as you can see in the panel B. Not all of the crypts fluoresce, so we couldn't quantify exactly how pure our CD24 low was. We did find that the cells grew in clonal analysis. You can see here at day 0 single cells. And you can see them progress and form these enterospheres, which are basically just multicellular, essentially spheres that have a lumen in the center, replicating somewhat of a mini-gut. And crypts begin to bud out by day 14 all the way to day 25, and they become these multilobular, what we call enteroids. And we then section these and stain them for lysozyme, which is a marker of Paneth cells, substance P, which is a marker of enteroendocrine cells, sucrase isomaltase, which stains enterocyte, and MUC2, which labels goblet cells. So we've then now showed that these cells can replicate themselves and the 4 lineages, which is a definition of a stem cell. So what this does is it allows us to ask some very interesting questions. If we had the stem cell cycling, can we go to the tab prior one? And one back, actually the cycling ISC. Thanks. So what this does is it allows us to take mouse and investigate what the stem cells are doing, because we can get the CD24 population and evaluate it with a secondary measure. And here we're using EDU. This is a thymidine analog, which is incorporated into the DNA, and we can then bind a dye to it. So in the same way that you would use BRDU or tritiated thymidine, we use EDU. And the real name is way too long to say. So, but it's a great kit made by Invitrogen that we've used in flow cytometry. So what we do is we inject this mouse an hour ahead of sacrifice. We then make a cell preparation, and we then evaluate with CD24 and EDU. So we can see what portion of these stem cells are cycling by flow cytometric analysis. And this gives us a great tool to look at factors that affect ISC activity. We can go to the next slide. One of the things that we're working on right now is looking at how the intestine regenerates following damage. And we know that it's very resilient. It can be almost completely wiped out, and if the stem cell compartment in the Crypt base is intact, it can regenerate the intestinal epithelium. And here what we're looking at is tissue in an untreated mouse and 4 days after a prescription drug that has cytotoxic effects. And what you can see is that the Crypt depth lengthens at day 4, so maximum damage is at about day 2, and then these crypts begin to increase in length as the stem cell pool expands and as the stem cells begin to divide more rapidly. To measure that, we can use EDU in combination with CD24 so that we're looking at the stem cell zone, and we can ask how many of these cells are cycling in an untreated mouse and how many are cycling at day 4 during this regeneration phase. So here, these are cell preparations from a control mouse. And the first plot we're looking at is, or the top panel rather, is EDU only. And this is a fluorescence -1 control that we run. So it's the same cell prep in the very first panel that you would see in the side population. It's just a heterogeneous mix of intestinal epithelium. We then select for singlets. We then do another singlet selection using side scatter area by side scatter linear. And we carry that forward onto a plot with CD24 on the y-axis and EDU on the x-axis. And here you can see the EDU positive cells pushing out along to the right-hand side of the x-axis. And we can set our CD24 low gate down on top of the negative. It's shown here in green. And the pink box is highlighting the portion that will be CD24 low and EDU positive if that is to occur, if they do shift up and they are EDU positive. And that's what Juan's pointing out right now with the red marker, is this is the CD24 or CD24 low EDU positive fraction. So these are intestinal stem cells from the Crypt base that cycled during that 1 hour pulse of EDU. And so what we can do now is look at mice that have received this drug that has cytotoxic effects. And here we're looking at these cells at day 4 after these mice have been hit with this drug. And what we hypothesized is that we would see an increase in the number of these rapidly cycling stem cells. And so here again on the top panel is the EDU only control. You can see a little bit of autofluorescence, probably from the fixation and permeabilization, but it's somewhat negligible. And then we run the sample with our gates established, pre-established. We run the sample that has both CD24 and EDU in it. So this data may be a little bit confusing to you. It would be to most people that don't look at it very often. And so let me show you. This is again, so here we're looking at the bar chart of the data. So this is a great way to convey this data rather quickly to somebody that's not experienced with flow cytometry or even just the tissue or experiment you're working with. So while this looks very familiar to me, in a flow plot, if you're giving a 3-minute talk or even a 10-minute talk at a conference and you have to convey this information, it's somewhat hard with a dot plot to get the point across fast. And here you can see that in mouse 1 and mouse 2, we actually had a pretty small number of cycling intestinal stem cells, roughly 18 to 19%. During this regeneration phase, we see a bump up to probably a mean of 42%, 43% actively cycling, and you can see them represented here, panel on the left. And what's key is that these calculations are being made. We're asking what percentage of the CD24 low is cycling. So we're saying what percent of the CD24 low EDU positive is cycling from the entire CD24 low. So it's kind of a 2-layer problem. Typically, when we look at the stats on a flow plot, as we move the gates, it gives us a number of, this population is this percent of all events. But here we're asking about what percentage is this gate from this other gate. And that's what these custom tokens allow us to do. We can ask more complex questions. And typically I would have to, here one's showing you that this green gate is all of the CD24 low, and the pink gate is just the CD24 low EDU positive. So we're asking what percentage is the pink gate of the green gate. And as the gates move, the bar chart moves, and that gives us a way to get these stats quickly. Otherwise, with the program that I was using prior, I would have to hand tabulate these calculations, and then if I decided to move a gate, let's say I made it more stringent towards singlet, I would have to retabulate all my stats. And so if you're doing multiple biological replicates, it can really slow you down. And then, with this bar chart here, I think the really nice thing to do is to be able is that we can quickly convey this point. Just glancing at these bivariate plots, it may not be so convincing. I think it would be a great to accompany the bivariate plots with the bar chart, because obviously we don't want to mask any data or anything on the plots. But for somebody to kind of quickly flip through a journal and see a panel including both, they can look at the bar chart and get the message. So I'm really excited to try this on our data. We only have this NF2, but it's something we're working on, and I'm having a lot of fun with these custom tokens. It gives you as much complexity as you want with regards to calculations, and so hopefully I'll be fluent in this soon. And I'll hand it back to you, Juan.
Thank you, Rich. That was awesome. All right, so about 1:55 here on the East Coast, so I think at this point we're going to start wrapping things up. If you have any questions, please feel free to enter the question into the questions section on your webinar control panel, and we'd be happy to answer them for you, or we can just take them offline and we can have an offline discussion regarding the answer to your question. But here I want to summarize basically what we've seen overall and how FCS Express can be useful in the type of analyses that we've done. So overall, FCS Express provides a convenient all-in-one package for your flow cytometric needs. So all the presentation that you've seen so far has been done exclusively in FCS Express. There's no PowerPoint involved. It's a combination of slides versus with flow data. So as we go through each of these pages, we could insert pictures, text, just like you would in PowerPoint, and we can also do our flow analyses all at the same time. So just like with any flow software, you can move your gates, all your information updates automatically, and if you're viewing that information on a bar chart, as you saw before, the bar chart will update automatically as well. FCS Express provides a very convenient way to streamline your workflow with a simple and easy to use interface. As you saw earlier with the stem cell enumeration assay, we can include all these different types of tokens in there where we can focus on the stats that are relevant to the type of analysis we're doing. And we can also incorporate a lot of drag and drop capability to really make that easy. As you saw where I can drag and drop that canned comment, we can also do the same in the creation of tokens or custom tokens where we can just click and drag them to a field, and we can just tell the software what events come in specifically from a particular gate or from a particular plot that we want to include in that token. And most and foremost, FCS Express offers a number of ways to make your point. I think that was most evident from the use of the bar chart. We also have something called the scatter chart. Just like you would create a scatter chart in Excel using a line graph, we can do the same here in Excel using the same type of information. Instead of inserting a bar chart, what we do is we insert a scatter plot. And we have the ability to connect the lines just like you would in Excel and formulate the type of data that you need to get your point across. Absolutely. Also, we have a number of resources available to our customers in the form of online tutorials and reference manuals that are readily available on our website. And we also have discussion forums that are available on our website where people like yourselves come in, ask questions, and they get responses from the Novo software team. So if you haven't already, please feel free to visit our site at www.denovosoftware.com to download a free 30-day demo. If you have any questions, please feel free to email us at support@denovosoftware.com. We'd love to hear from you. Many times we'll respond within 24 hours, if not a couple hours after you send us the email. So if you have any questions, please feel free to enter them at this time. And we'll just take a couple minutes to see if any questions come across. Otherwise, thank you for joining us today. We look forward to hearing from you, and we look forward to seeing you take the software out for a test run.